Hi everybody, my name is Joao, and today I'm gonna talk to you about transcript levels and sequence determinants of protein abundance and noise in E. coli. So our general aim can be described as to create an integrative and quantitative model that could map genotype to phenotype. And more specifically, we are interested in understanding what are the determinants of protein abundance regulation genome-wide in E. coli. What this means is that we really want to understand how do we go from DNA to messenger RNA levels, and then from messenger RNA levels to protein levels. And this is particularly important because although we can measure messenger RNA levels at relatively high throughput, protein abundance quantification methods are lagging behind. And moreover, transcript abundance alone can only explain 50% of total variability of protein abundance genome-wide. So this raises the question of what are the determinants of post-transcriptional regulation. And to answer this question, I considered multiple sequence features that would be responsible to control translational efficiency in E. coli. And these multiple sequence features come mainly from two different regions, and which I show up here, and that would be the translation initiation region, or the TIR region, which considers the 5' UTR of the transcript plus the 5' end of the coding sequence. And the idea is that this region alone should be enough to encode all the translation initiation regulatory signals. And here I access multiple sequence features such as the accessibility of this region due to the RNA structures that can be formed, and also like the affinity between the ribosome and the Shine-Dalgarno signal, etc. And the second region, which is the coding sequence region, contains other regulatory signals that are supposed to control the translation elongation rate, including things such as the codon usage or the amino acid usage. And the goal here was that starting from a very large number of more than 100 sequence features, as well as experimental data on RNA and protein abundance, we apply the feature selection procedure to derive a quantitative model that has maximum predictability but minimum complexity, that is, as fewer variables as possible. And we also assess these methods using cross-validation procedure. And in this case, we are just using partially square regression analysis to predict protein abundance as a function of the multiple explanatory variables. So our model, which only contains 16 predictors, is able to explain 66% of protein abundance variability genome-wide. And what I'm showing here is just a plot of the predicted abundance versus the observed abundance. And I call your attention for the fact that we are actually being very quantitative and predicting average abundance molecules per cell. And as you can see, our cross-validated R-square, it's 0.65, which is very close to the R-square that we get when we use the entire training set, which indicates that our model does not suffer from overfitting. So interestingly, our model suggests that messenger RNA levels alone can explain about 53% of the total variability in protein abundance, whereas regulatory signals encoded within the coding sequence can explain 12% of the variability, and regulatory signals within the translation initiation region can only explain 1% of the variability. Moreover, because we use the linear regression model, we can look at the regression coefficients and get an idea of the relative importance of the multiple predictors. And these are the final 16 predictors. And we have like messenger RNA levels that we have just one feature that comes from the translation initiation region, whereas the remaining 14 features come from the CDS region and mostly reflect codon and amino acid preferences. So the data set that I was actually using to build this model is also based on around 1,000 strains, where each strain is tagging a different gene with the YFP gene, and was developed in this study here. And what this allowed the authors to do was to use automated imaging to quantify all the proteins, or at least like 25% of the proteins in the E. coli proteome. And what I'm showing here is just an example where the protein ADK is tagged, and as you can see, there is a lot of cell-to-cell variability in the protein abundances that you can see. And what the data set provides you is not only the average protein abundance that you get for each protein, which in this case it would be around 600 copies per cell, but it can also tell you how much this protein abundance is going to vary across the cell population. And this is also known as the expression noise. And the very popular metric of expression noise is just the standard deviation over the mean of protein abundance across a cell population, which I'm plotting here on the y-axis. And as a function of the average protein abundance for a given gene. And as you can see, there is this strong negative dependence between these two metrics, where the noise will decrease as the average abundance of a given gene increases. And each dot in this plot is just a different gene. And what this orange line means is just the median expected noise for a given average protein abundance. So what we did next was to remove this effect between noise and protein abundance, was just to calculate what is a metric which is called the noise differential, which is basically the distance between the noise of a given gene to the noise that you would expect for that particular average protein abundance. And this gives you an estimate of how noisy that gene is in comparison with other genes that are expressed at the same level. And what we did next was to use our estimates of transcription and translation efficiency from our previous model that I just showed you, and see how this would affect the noise differential of each given gene. And what we found was that transcription and translation efficiencies have a similar magnitude effect, and even though they have like opposite inverse effect, which suggests that like for a given gene, you could just tune the expression noise of that gene just by playing with the transcription and translation efficiency, independent of the average abundance that gene has to have. So just in summary, we have developed an integrative model to quantify the determinants of protein abundance on a genome scale in E. coli. And the model suggests that protein abundance is primarily determined by the transcript level and by effectors of translation elongation, whereas just only a small portion of the variation is explained by translation initiation. And we also show that transcription and translation efficiency may have a similar magnitude but opposite effect on expression noise. And with that, I would just like to thank my advisors, Adam Markin at Berkeley and Miguel Rocha at Minho, and as well as all the funding sources. And thank you for your attention. I would be happy to take any questions you might have. Hector.
Excellent talk, Joao. I was wondering if you have any wild hypothesis on the origin of that 34 of an unexplained variant that you would like to share with us.
So, one of them immediately is the experimental error. So the experimental error is, for this particular data set, is supposed to explain around 14% of protein abundance variability. So that's certainly something that we cannot explain. And in our model, we are only also considering transcriptional and translational and canonical translational regulation. There are other types of regulations that we do not take into account, like protein degradation or other kinds of translation regulation that are not picked up by the initiation and elongation features that we consider, and we think that those could at least improve the prediction power of the model and hopefully take it to the 100%.
I have a question about the data you use to determine the abundance of RNA and proteins. So can you elaborate more on that? And did you use any bias correction methods to correct the abundance estimates before applying your model?
In protein abundance?
And RNA abundance.
Okay, so protein abundance, we use that fluorescence tagged strains, and what we use as the average abundance is just what is reported in the manuscript, and it's like basically the average abundance across the cell population. And this, I think that's pretty unbiased. And regarding RNA, messenger RNA abundance, we use RNA. So the authors of that same paper used RNA-seq to quantify that the messenger RNA levels, and in that paper they show using like complementary assays, such in the case of RNA abundance, RNA fish, and they show that their RNA-seq measurements match well with those measurements. So I think. And they all do all these bias corrections for RNA-seq.
Thank you.
You're welcome.
All right, if there are no other questions, we'll thank Joao again. Thanks.